Hello everyone, myself Dr. Ashwath Narayan. In this session, we're going to discuss about heritability problems. Before going to the heritability problems, we need to know about how this heritability is linked to polygenic variance. And what is polygenic variance and other factors will be discussed in this video. First of all, we need to proceed with a topic called as polygenic inheritance. What is polygenic inheritance means? It is also called as quantitative inheritance. It is also called as a multifactorial inheritance, where multiple genes, independent genes, which are present in non-homologous chromosome, which shows additive gene effect on particular phenotypic character. Before going to polygenic inheritance, we need to know whether it is allelic or non-allelic gene interactions. You already know, if you are familiar with genetics, you clearly know about allelic and non-allelic interaction. What is allele? Allele is alternative form of gene which is present on homologous chromosome which code for some phenotypic trait. Take example here, tall is there, dwarf is there. Both are, both are present in homologous pair. Tall, tall gene and dwarf gene is there, which is also called as allele, which code for plant height. Right. So like that, alleles which are present on homologous pair, homologous chromosome, which code for some phenotypic trait. But in this polygenic inheritance, this is called as non-allelic interaction. Non-allelic means you already know that. So alleles, it is the genes which are present in non-homologous chromosome. Non-homologous means assume that this is metacentric and this is acrocentric chromosome. Here A gene is there, here B gene is there, which is not at all homologous, because but this A gene and B gene will show some features, some effect on some phenotypic trait, which is mainly shows effect on plant height. That is called as non-allelic gene interactions. In that, more than three genes, more than three genes, more than four genes, you can observe that they show effect on some phenotypic trait is called as polygenic inheritance. Why it is called as quantitative inheritance means it shows additive gene action or cumulative gene action. We're going to discuss that in next slide. Some characters are there with respect to polygenic inheritance we need to know about first, before proceeding to actual problems of heritability. So polygenic inheritance will have additive gene effect. Single gene contributions are there which shows additive gene action. Assume that for example, skin color in human being is a classical example of polygenic inheritance. In that, the extreme melanin content person is called as negro, and then without melanin is called as white. In between, many other phenotypic traits are there. When negro, assume that negro genotype is capital A, capital A, capital B, capital B, and white genotype is small a, small a, small b, small b. If they cross, they produces intermediate phenotype called as mulatto. Right? This is called as mulatto. Right? If mulatto is self-crossed, right, it produces phenotypic ratio in the in the form of the numbers here, 1 is to 4 is to 6 is to 4 is to 1. In that, 1 is called as negro, 4 is called as black. Why? Because because of 75% melanin. Here negro is 100% melanin, and mulatto, you already know the intermediate 50%, and then 4 is called as light, and it contains 25% melanin, and white, it contains 0% melanin. Understand? Here each gene contribution will be calculated, and each gene contribution is counted, and to detect that will be a very difficult task that will be detected by some statistical analysis. So here one of the trait is multiple genes produces equal effect. You can see here, assume that it is, some plant is there and it has genotype AA, BB, and because of this genotype, it produces 9 inches of carb length. And one more genotype is there, small a, small a, small b, small b. It gives cumulative effect of 3 inches. In that, number of genes involved is 4, and you need to divide by 4, and it will show 2.25 or something. So inches, it is the contribution of from the single gene. So single gene produces contribution of 2.2 inches, and cumulatively it gives 9 inches, right? That will say each gene produces equal effect, and and also shows cumulative effect. That means 2.25 plus 2.25, like that. You need to sum up that 4 times and you will get 9 inches. That shows additive gene effect or cumulative gene effect. And this polygenic inheritance is not at all compared with multiple allelic interaction. Why? Because this is allelic gene interaction, and polygenic inheritance is non-allelic. Non-allelic means they are present in distant chromosome, and I know code for some phenotypic trait. That's it. Here alleles is different, and polygenic inheritance is non-allelic interaction. And one more point is there is no linkage and dominance. As I said earlier, each gene contribution is equal. Because of that, there is no dominant allele, recessive allele in this genic interactions. One more thing is no linkage. No linkage means, as I said earlier, linkage only present in allelic gene interactions, where alleles are the alternative form of genes which is mainly present in homologous pair. Their linkage effect will be there. But you can see here, this is metacentric, this is acrocentric chromosome. Here one allele is there, one gene is there A, here one gene is there Z, and both are interacting. That there is no chance of linkage, right? Because of that, the polygenic inheritance is not at all affected with linkage here. And it shows continuous variation. As I said earlier, it shows when Negro marries to white, and it produces mulatto. And mulatto is self cross. Then it produces the genotype in the ratio of 1 is to 6 is 1 is to 4 is to 6 is to 4 is to 1. One is Negro. If you plot a graph, it shows continuous variation. Continuous variation like this. It shows a parabolic or bell-shaped curve. And the genic interaction is very complex, and it will be difficult to predict the phenotype externally. But if you go with statistical analysis, it is easy to calculate that. Next, we move on to example for polygenic inheritance. In that, one of the example is skin color and pigment. We already discussed about it. Here number of genes will be more, but for I showed a simple example. 1 is to 6 is to 4 is to 6 is to 4 is to 1 is simple example. But if your number of genes are increased, then the ratio will increases like this. 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to 1, like that. The ratio will goes with the increase in genotype. And then human height, also one of the example which is inherited by polygenic interaction. Okay, here human height means one is 6 feet tall and 5 female is 5 feet tall. Then there will be intermediate height will be produced. And then one more thing is there is no mandation that if 6 assume that this is the genotype of the man, this is genotype of the woman, and it produces intermediate plant height, human height, like that. The he is 6 feet tall, she is 3 feet tall, and the number of genes are 4. Like divide, and it will you will get it's 9 inches, like that. If 9 inches is there or 3 inches are there, if divided in gene contribution like that, and it shows some phenotypic trait, it is not mandation. Why? Because here environmental factors also work here. There is a major controlling entities where the environmental factors also work here in deciding the phenotypic traits here. Because of that, in human height, there is no mandation that it will shows intermediate height of human being when tall man mated with dwarf woman. There is no mandation. Why? Because some environmental factors are there, nutrition is there, and other factors are there, hormonal actions are there, and different factors are there that will decide the height of human being. Next one is human eye color is also inherited by polygenic inheritance, where black eye color and the blue eye color is crossed. Then it will shows the ratio in the 1 is to 4 is to 6 is to 4 is to 1. Like that, black eye, dark brown, light brown, green, and blue eye is there. Light brown is intermediate color. So this is the example with human beings. With respect to plants, kernel color in wheat is classical example, and the polygenic inheritance is primarily identified in kernel wheat color. Kernel color in wheat, is the first experiment conducted and identified polygenic interaction. It is also called as QTL or quantitative inheritance. In that same, dark red wheat is crossed with white. It shows intermediate color in F1 generation, and F1 itself, it produces the ratio 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to 1, like that, if number of genotypes increased. And length of the corolla in tobacco is also one of the example with respect to polygenic inheritance. We know long corolla, short corolla, and intermediate corolla is classical examples here. And one more thing we need to, we skipped out in previous slides is effect of environment in polygenic interaction. This is very crucial. Why? Because the environment mainly affect the polygenic inheritance here. If environment effect is more, that heritability inheritance of the trait will be very difficult. So that is the core point of heritability. Whatever we discussed about polygenic inheritance is mainly affected by environment, and the effect of environment will be calculated by a term called as heritability. Heritability means, by definition, how much the genotypic trait will be inherited, and that will shows in phenotypic trait. Right? Whatever phenotypic trait is mainly influenced from the genotype, or how much quantity of genotype will be expressed in next generation will be calculated through the formula of heritability. Right? Heritability, as I said earlier, heritability refers to proportion of observed variation with respect to the genetic variation which attributed by individual plant. That is called as heritability here. Heritability is of two types. One is called as narrow sense heritability, and second one is broad sense heritability. Both heritability is mainly influenced by environment and genotype, that will be discussed in next slide. And narrow sense heritability is indicated in small h square, and broad sense heritability is indicated in capital H square. And both values will vary from 0 to 1, and if the value is near to 0, then environmental effect is more. When the value is near to 1, then gene effect is more. So environmental effect is more when the value is near to 0, and gene effect will be more when the value is near to 1. Next, the types of inheritance. In that, the first one is called as broad sense heritability. Broad sense heritability is the type of heritability where you can observe that it shows cumulative effect of both genic variation as well as environmental variation. So that means, cumulatively genic variation plus environmental variation which gives the phenotypic variation. So whatever phenotypic traits are decided in particular plant or animal will be decided by both genotypic and environmental effect. So the formula is Vp is equal to phenotypic variation is equal to the cumulative effect of genotypic variation plus environmental variation. And the formula for broad sense heritability is H square. Capital H square is equal to Vg by Vp. That means it is the ratio between genotypic variation divided by, and with respect to phenotypic variation. Genotypic variation and phenotypic variation, that will gives the broad sense heritability. Capital H square value, if it is near to 0, so here the environmental effect is more, and no genetic influence is there. That means whatever temperature, sunlight, humidity, and other factors are there that mainly influence on the particular phenotype, and no genetic effect influence will be calculated. When if a plant or animal shows the narrow sense heritability, the broad sense, pardon me, broad sense heritability near to zero, then to improve the quality of a breed or a particular plant, you need to go for change in the environment. If you change the environment, definitely you'll get good yield here when the broad sense heritability is near to zero. When the broad sense heritability h square value is near to 1 or 0.9 or 0.7, definitely there is more of genetic influences there rather than the environmental influence. There is no environmental influence will be there. That means, lesser environmental influence is there when the value is near to 0.9, 0.8 like that, but more of genetic influence is there. So in that case, you need to go for artificial breeding or conventional breeding in selecting good yielding plant here. In case of h square value is near to 1. So what is the chances when h square value is 0.5, sir? Is near to 0.5? Definitely there is influence of both environmental factors as well as genetic factors will be there. In that case, we need to select either we need to change the environment first. If the yield is not expected, then we need to go for genotypic variation. So that is the conclusion in case of broad sense heritability. So here, we need to proceed with further elaboration of this formula. H square value is equal to Vg divided by Vp. So Vg is again elaborated. Genotypic variation is also included. That means, it is a cumulative effect of additive gene variation, dominant gene variation, and genic interaction. Both the three, all of three will comes under genic interaction. Whereas phenotypic interaction or variation, which is the cumulative effect of additive, dominant, genic interaction as well as environmental interaction. So the formula which is indicated like this: Vg is equal to Va plus Vd plus and Vi, whereas Vp is equal to Va plus Vd plus Vi plus Ve. So here additional environmental effect also there with respect to phenotypic variation. So if broad sense heritability is near to 1 or 0.6, assume that the value is 0.6, does not mean that 60% of the corn size is mainly determined by genes and 40% of the corn size is determined by environment. The actual factor is 60% of corn size is whatever size is observed is due to genetic differences. That's the conclusion you need to give. That means, if the value is 0.6, that means 0.60, then definitely 60% corn size is not determined by genetic effect, but it is mainly observed by the 60% is mainly observed due to genetic differences here. If the heritability is broad sense heritability is 0.2, definitely 20% is mainly affected by environmental variations here. That means whenever the broad sense heritability is 0.6, then you need to go for artificial selection in getting good yielded plants or animals. Where heritability is 0.2, then just simply you can change the environment and you will get good yield here. Understand? So in that, h square value will give complete information whether you need to go for change in, you will go for changing environment and good yield, or you need to go for artificial breeding in getting good yield with respect to animal breeding and plant breeding. Next one is narrow sense heritability. Narrow sense heritability is not at all focusing on all the genetic variation, genetic variation, dominant variation, and other additive gene variations. They are not. Sorry, it's not additive gene variation. There is epistatic variations here. And I can observe that narrow sensitivity is not at all depends upon that. It mainly focus on additive gene variation. What is additive gene variation means? I can observe that. For example, take example here. A mean cob length. This is the cob length of corn. Here 6 centimeters are there. 6 inches. Sorry, 6 inches is there here. In 3 inches are there in other plant. If both are mated, both are crossed, it will shows intermediate cob length here in F1 generation. If F1 generation is self, then it produces plenty of F2 plants here. In that, whichever phenotypic traits are there, which resembles the grandparents, that shows additive gene action. That means the additive gene variation is a variation which is observed whenever in F2 generation, whatever plants are there, which shows exactly whatever, which resembles exactly whatever the grandparental phenotypic traits are. That means whichever shows 6 inches in F2 generations will shows additive gene action, where the additive gene effect, which mainly resembles the phenotype as same as of the grandparents. That is called as additive gene action. In that, the narrow sense heritability is calculated by small h square divided, is equal to uh, the ratio between the variation of additive gene variation with respect to phenotypic variation. So here also, if the narrow sense heritability is near to 0, then the environmental effect is more and no additive gene actions will be there. Where the value small h square value is near to 1, here no environmental influence is there, but here more of additive gene actions are there. And then if the value h square value is intermediate, then both additive gene action as well as environmental effect will be there. That is the conclusion with narrow sense heritability. So students, now we will move on to actual problem related to heritability. In that first problem is phenotypic variance is analyzed for yield in rice plant, and following variance components are obtained. VA, VD, VI, VE is given here, and find out narrow sense and broad sense heritability. You already know the formula, right? Capital H square is equal to Vg by Vp, and you already know what is Vg means. It's a cumulative effect of genotypic it's a cumulative variation of additive gene variation plus dominant gene variation plus genic interaction variation. And you already know the Vp, that means Va plus Vd plus Vi plus and Ve. So by summing up these values, you'll get that means 3.5 plus 2.3 plus 1.9 plus 1.9 divided by again 3.5 plus 2.3 plus 1.9 plus 4.5 4.7, sorry, that is equal to, so numerator you will get 7.7 and denominator get 12.4, and you will get the value of 0.62. So if h square value is 0.6362, definitely the value is more influenced by genic interactions, and less influenced by environmental interaction. And we need to calculate narrow sense heritability here. We calculated h square value, right? So then narrow sense heritability is equal to small h square is equal to Va divided by Vp. So what is Va? Va is 3.5 divided by Vp is 12.4. So total value, this h square, small h square value is equal to 0.28. That means here h square narrow sense heritability is 0.28. That mainly indicates that it is mainly influenced by the narrow sense heritability is mainly influenced by environment, and no additive gene effect will be there. That is the answer for first question. So next we will move on to next question here. The Narrowson's heritability of body weight in mouse is 0.7. Body weight in mouse is 0.7. The phenotypic variation VP is 0.6. What is the additive gene variation for the body weight in mouse? That means what they given Narrowson's heritability small h square is given 0.7 Narrowson's heritability, and phenotypic variation VP is given, that is equal to 0.6. We need to calculate VA, right? We already know the formula, h square is equal to VA divided by VP. But what we should calculate? VA should be calculated. VA is equal to h square into VP. That is equal to h square is 0.7 into VP is 0.6. Then the value is equal to 0.42. VA is equal to 0.4. That's the answer. Next, we'll move on to one more problem here. A scientist conducted an experiment in mung bean. He raised 10 different varieties of mung bean in a field and counted the length of the pod. The variance for this character is 15. That means, scientists grow different varieties of mung bean in the field, and it shows, in the field it definitely shows phenotypic variation that is equal to 50. He then selected a single variety and brought it to greenhouse and bred for 2 years. He again raised the second of the, seeds of this variety in same field condition. The variation showed is because of change in environment. PE, the variation showed is 8. From the data, find out Dawson's heritability. You need to calculate capital H square value. So you already know capital H square is equal to Vg by Vp. Before that, in one more formula is there. Vp is equal to Vg plus Pe. So Ve we know and Vp we know. We need to calculate Vg. Vg is equal to Vp minus Ve. Is equal to 15 minus 8, and you will get the value Vg is equal to 7. Vg is equal to 7. Then apply that h square value here. We calculated Vg here. Vg is equal to 7 divided by Vp 15, and we will get the value of 0.42 or 47. Sorry, h square is equal to 0.47. That is, it is near to value of intermediate. That means both genes as well as environment is influencing on it. Environment is influencing. Next, we will move on to one problem here, one more problem. The narrow sense heritability and broad sense heritability of wool length in sheep are 0.2. Pardon me. 0.2 and 0.6 respectively. The narrow sense and broad sense heritability of wool length in sheep are 0.2 and 0.6 respectively. The additive gene variation is 5. Find out Vg, Vp and Ve. That means small h square is given, value is 0.2. Capital H square is given, value is 0.6, and Va is given, that is equal to 5. Right? So we need to calculate Vg, Vp and Ve. Right? So first the value is we need to calculate small h square. You already know the narrow sense heritability small h square is equal to Va divided by Vp. Right? In that you already know h square value and Va value, Vp value you need to calculate. So in that, Vp is equal to Va divided by h square. That is equal to VA is equal to 5, and H square is equal to 0.2, and VP value is calculated like this. VP is equal to 25. VP is 25, right? And then VP is calculated, and we need to calculate VG here. VG, and to calculate VG, again we need to go for formula here. Capital H square is equal to VG divided by VP. So in that we can calculate VP here. Why? Because we already know the value of capital H square, and we know the value of VP here. We need to calculate VG is equal to H square into VP. That is equal to H square. VG is equal to H square. H square is capital H square is 0.6 into uh, VP. VP is 25, and in that VG is equal to 15. Right? VG is equal to 15. We calculated VG, VP, and we need to calculate VE. So VE is equal to, you already know the formula. You already know the formula. VP is equal to VE plus VG. In that we need to calculate now. We need to calculate VE now. So VE is equal to V in VP minus VG. Then the value is 25 minus 15, that is equal to 10. VE is equal to 10. So we calculated VE, VP, and VP, and VG is calculated here. VP, VG, and VE is calculated here. Understand? So VP, VG, and VE calculated. That's the answer for that. Next, we will move on to one more problem here. The mean grain yield per plant of wheat grows in field of Bangalore was 12 grams, and what is that given? Mean grain yield per plant in wheat in the field means you need to go for VP. VP is equal to 0.23. The variation showed is 0.23. When grows in greenhouse, in condition, in control condition, only genotypes work, because you already take over the control with respect to environment. Because of that, whatever variation which is uh, comes whenever grows in greenhouse. When grows in greenhouse under controlled uniform condition, the mean grain yield per plant was 12 grams, and the variation was 0.07. That variation is 0.07. That is VG. And estimate broad sense heritability. We need to calculate capital H square here. You already know the formula. H square is equal to VG by VP. And now VG is equal to 0.07 divided by 0.23. It's a direct question here. 0.30 is capital H square. So the value is 0.30. That means you already know the conclusions here. If the value is near to 1, near to 0, more environment is very environmental things are influencing on phenotypic trait. If it is 1, definitely genes are influencing more as compared to environment. If the value is 0. Intermediate value is there, both environment and genes are influencing in deciding phenotypic traits here. Here the value is 0.3, then definitely more environmental things or environmental factors are very show the influence on that phenotypic right here. Next we'll move on to the problem. Next problem here. The variance of two inbreed line of bajra. The variance or variation of two inbreed lines of bajra for year length are 14.5 centimeter square and 13 centimeter square. In random pollinated population, the variance was 2.9. centimeter square. Estimate broad sense heritability of ear length for this population. So, if some confusions will be there for the people, how to how to know what is VE, what is VA, what is VP. In that, you need to calculate broad sense heritability. What is given here? The variance for two inbreed lines of Bajra. That is field, right? Field means that may definitely that is called as VE, environment. So that is two values are given. You need to take average 14.5 plus 13 divided by 2. You will get the value of 13.75. That is VE value. VE is equal to 13.75. Then one more variance is given. The variance, random pollinated population. The variance means that phenotypically that is given. VP is equal to 29.7 centimeter square. Then you need to calculate broad sense heritability. So before going to that, we already know P is equal to VE plus VG. So here if you calculate VG here, VG is equal to PP minus VE, that is equal to VP is 29.7, VE is 13.75. The VG value you will get is VG value you will get is 15.95. After that, you need to calculate broad sense heritability. H square is equal to Vg divided by Ve. Then Vg is equal to 15.95 divided by Ve 13.75. So the 29. Sorry, Vp. Sorry, Vp. Sorry. Pardon me. It is Vp, not Ve. Vg by Vp. The value is 29.7. 29.7 here, and if you calculate that, so the value will be 0.54. So if broad sense heritability is equal to 0.54, definitely. So it is both influenced by environment and genes here. Next, we will move on to the last problem in this section. In flock of sheep, the narrow sense heritability of wool production is 0.7. Its phenotypic variation is 73 kg square, genetic variation is 55 kg square, epistatic variation is 2 kg square. Calculate dominant genetic variation, that is VD, you need to calculate dominant, and environmental VE you need to calculate. Understand? So what is given to you? Narrow sense heritability is given, small h square is given 0.7, and its phenotypic variation is given, VP is given, that is equal to 73 kg square, and genetic variation is given, VG is given 55 kg square, and epistatic variation, what is I, genic interaction variation, is 2 kg square. Then you need to calculate VD and VE. So in that, first we need to go for the formula h square is equal to small h square VA divided by VP. Then VB value, VH square value, you need to calculate VA value, that is equal to H square into VP. H square is equal to 0.7 and VP is equal to 73. So in that, VA value is 51.1 VA value. We calculated VA here, and next we'll proceed to one more formula is equal to VP is equal to VE plus VG. So here we already know VG here and VP here. So we need to calculate VE here. VE is equal to VP minus VG, that is equal to VP is equal to 73 minus VG is equal to 55. Then you will get 18, that is equal to VE is equal to 18. So we calculate VE now. We'll move on to the one more formula, VD, to calculate VD here. So you already know that VG is equal to VA plus V dominant variation, and then VI genic interaction, right? You already know VA here. VA is equal to 51.1. We don't know VD here, and we already know epistatic variation, and VG we know already. VG is given here. Then VD is equal to you'll get, if you calculate this one, you'll get around 1.9. So VA we already know that, VE 18 we calculated, and VD calculated. This is the answer for this question.